Hi, everyone. My name's Josh. I'd like to start by acknowledging the traditional custodians of the land on which we sit today, the Gadigal people of the Eora Nation. It is upon their land that we make the medicine, as my 5-year-old likes to describe what we do here at Psylo. So I'm going to start with a quick overview of what we're doing, how we're going with partnerships, and some people who are getting involved with this project called Psylo. I'm then going to hand over to my main man, Sam, who's going to go through a lecture on Psychedelic Science 101. And then we're going to go through a panel discussion on TGA and rescheduling, potential rescheduling of psilocybin and MDMA. A decision on that is imminent. I've been refreshing my browser all day today. The TGA said they were going to make an announcement this week. May bleed into next week. We'll see. So to start, we were really excited about our pre-seed round of funding, and thank you for those of you who participated in that. I wanted to put that in the context of the 1.1 billion US dollars which have flowed into psychedelic startups in the last 2 years. So there's a lot of hype and a lot of money flowing into this space. Most of this money is going towards clinical trials. So this is a slide from a deck that Valhalla Ventures put out a couple of weeks ago. It's actually a really good deck, and I highly recommend everyone go and take a look at it. So these are all companies that are progressing clinical trials on naturally occurring psychedelic molecules, such as psilocybin, DMT, and 5-MeO-DMT. All of the logos at the top are progressing those molecules through for treatment-resistant depression, okay? So the reason why we can go straight towards clinical trials with these molecules is because there's thousands, if not tens of thousands of years of history of consumption of these molecules. So this is great work. It's quite expensive work. I'd say 80% of that 1.1 billion is going towards these clinical trials. You can go straight into humans, and a lot of people will get medical solutions from these molecules. This is not what we're doing. What we're doing at Psylo is we're focusing on developing psychedelic-inspired NCEs. So I'll unpack that a little bit by starting with the acronyms. Life Sciences, by the way, has many acronyms to offer. I'm only going to go through 2 of them. NCEs are new chemical entities. If you read our monthly updates, this is the term that we refer to the most. These are molecules which, as far as we know, have never existed in nature or otherwise. CROs are contract research organizations. These are partners that we work with to synthesize and screen the molecules that we design. So Sam and I have launched our program in a virtualized model using these partner CROs to develop NCEs, which are psychedelic inspired. So I'm going to explain a little bit about what that process looks like. So the first step in developing a research pipeline in psychedelic NCEs is Sam sits at a computer and he designs novel molecules using software. Okay? So basically he's taking the molecules of psilocybin, DMT, 5-MeO-DMT, and he's systematically tweaking them using MedChem tricks. And then we put those through a screening cascade to determine whether they have drug-like properties, whether they hit the appropriate receptors in the brain to be used as medicines in the future. So after we design those molecules, we then synthesize them with our partner CROs, okay? So basically this is bringing the molecules that Sam has designed into reality in the real world, into the physical world. So, when Sam and I get on calls with folks and they ask, okay, so this Psylo company, is this just, you and Sam? I like to say, well, it's me and it's Sam and it's the 30 chemists in China that report to Sam that are helping us to synthesize these molecules. So Sam is going back and forth with these chemists on the most effective route to synthesis for making these compounds. After they're made, we then screen them. So this is in vitro screening, is the first type of screening that we do. In vitro, like in vitro fertilization, is effectively in a cell culture. So what we're testing here is, do these molecules interact with the receptors in the brain that we're looking to target? If they do, we have a hit. We get really excited. Sam jumps up and says we have a hit, but then he sits down and realizes that he has a whole lot more work to do, because getting a hit effectively starts the process over in a way. Because basically the name of the game here is getting hits and then iteratively redesigning, synthesizing, and screening the molecules which interact with the appropriate receptors in the brain that we're targeting. All of the information that we,
How do you assess that it reacts with the receptors in the brain in vitro?
So you take the molecule and you dilute it at a varying degree of dilution, which is called concentration response curve, to basically, Sam,
He was doing such a good job. I didn't want to jump in and steal his thunder. Josh has learned an enormous amount about this process, and he kills it. So essentially, we take the molecule, we screen it against a cell culture, as Josh mentioned. This cell culture is an artificial system that has been genetically modified to express the human form of that receptor. The drug interacts with a certain pocket on that receptor to have an effect. There's sort of a signal cascade that happens as a result, and we use a fluorescent output to measure one of the results of that signal transduction. So it tells you that the drug is binding to the site, it's activating that receptor, and it's having a downstream effect. And we can quantify that because it's a light-driven readout. We can measure things and quantify the whole process. So we can rank all the different drugs in terms of their potency and efficacy and everything else. Hey, there you go.
In vitro, it actually changes the color perception.
We use a dye-based readout for this downstream effect. It's to do with calcium mobilization is the readout we're looking at, but there's a fluorescent dye that tells you how much calcium's been mobilized, basically. Yep.
How many different kinds of receptors are we targeting?
Ooh, this is good. This leads into my slides. So, no, I don't cover it in a lot of detail. There are 14 5-HT receptor subtypes. We're interested in 2 of those very specifically. We're very interested in not hitting one of them because it's associated with cardiac toxicity. And we think we might be interested in a few others because other drugs also hit some of these other targets, but we're not entirely sure. So currently we have a pipeline that looks at 3 subtypes, and we may move into another few from there.
So exactly like I was saying, that is how we conduct our screening. So all the information we get back from the screening and from the synthesis, we gather all this data, and this informs patents, which we file to protect the research that we've done, to ensure that we can continue investing in these molecules and drive them towards the clinic. So this is where we're at right now. We have designed 75 molecules. 71 of them are either under synthesis or have completed synthesis. 43 of them we have full CRC screening on. And we had a call this morning with our IP lawyers, and we are in the process of drafting our first provisional patents. We expect, thank you, yes. We expect to have 4 provisional patents filed before the end of the year. And for some context, the process for getting towards the patent stage is a process that can take years. And we've achieved it in a number of months. So that's why we're so exhausted, because the last couple of months have been quite busy. So just to give you a sense of what happens next. Next, we go into in vivo screening. That's testing our molecules in rodents. So that's something that we're going to kick off at the beginning of next year. We're currently getting quotes back from CROs for conducting that research. So that's our pipeline. Some partnerships that we've been working on. So we love the Commons, but we're actually leaving. We're going to UNSW, where we are setting up our laboratory. So Petra, thank you very much for putting us on the scent of UNSW. They are super keen to get us in-house, and they have pristine labs that were built in 2019, and we're super excited to get in there. CSIRO and ANSTO, both governmental organizations, both of them offer either matched funding or non-dilutive, just grants for conducting research with them. We've lined up partnerships with both, and we're going to initiate those as soon as we have our provisional patents in place. And then MRCF is the Medical Research Commercialization Fund. This is a government funding mechanism which is administered by Brandon Capital, which is a large biotech VC. And we submitted our application for their Curator grant on Monday, which is a $500K grant, non-dilutive funding with $500K of follow-on funding that's available. And we find out next week if we continue to the next stage. So fingers crossed on that. And then to wrap up, I'm really excited to talk about some of the people that have joined us at Psylo. So first, we've got Professor Danny Hoyer and Professor Laura Jacobson joining us from Melbourne. They're here today. Really excited to have them. So both are incredible pharmacologists. Danny from University of Melbourne currently, Laura from the Florey Institute, both joining us as a power couple of pharmacology. Super excited to have them. We've got Asher Brandt joining us as a computational chemist. Asher was not able to be here because he is in Saskatchewan. So he is a computational chemist, meaning he's going to help us use AI to discover novel chemical entities, something he can do remotely, and it's a part-time appointment, not full-time. And then full-time, we've got Will Jorgensen joining us as a med chemist, and we're super excited to have him joining the team this week. So welcome, Will. Welcome, Danny and Laura, and thank you all for coming. That's the end of my part, and I'm going to hand over to Sam to talk about Psychedelic Science 101. Thank you.
Just a quick question before you go.
Yeah.
You mentioned before that slide with all the logos on it. And they were all doing just clinical trials with like psilocybin and DMT or whatever.
Yep.
And all the money that's in the VC world is flowing into just those clinical trials.
Yep.
Whereas what you're doing is trying to find new chemical entities. Are there any others that are playing in that space?
Good question.
Very good question. So there are a few. Most of those companies have some form of NCE thing, but there are only a few that are really focusing on it. So there's Delix Therapeutics, there's Mindset Pharma, and who else am I missing? Bright Minds Biosciences. And Gilgamesh.
And that's about it.
That's it. Gilgamesh was also on that slide because they're looking to do clinical trials.
Why do you think the money is flowing into just the known entity, the known compounds rather than...
Because they're there and available, and because you can jump straight into clinical trials. You don't have to go through the in vivo, in vitro screening. You can literally dose tomorrow. Well, not
But
tomorrow.
if it's just a psilocybin, then everyone can do it, right?
Well, that's part of the thing. If you check out that deck from Valhalla Ventures, they talk about, okay, there's all of these companies that are chasing the same molecules for the same indications, and they're raising lots of money based on, excitement in the space and investor interest, et cetera. But, not all of them are going to be able to succeed. And also, because these are natural products, your ability to protect the sort of work that you do in that space is it's very hampered, right?
But their IP would be in how you grow it and how you harvest it.
Well, no, because most of them are using synthetic psilocybin. So, for example, Compass Pathways is one of the most advanced of these companies in terms of clinical trials for psilocybin. And their patents are around the method of synthesizing psilocybin, as well as things like the color of the walls of the room in which you're undergoing a psychedelic trip, and the hand holding.
Respect to them for taking a chance and trying to commercialize the space, because that's going to lead to a lot of other investment and excitement in the area. Their patents are around, they're not composition of matter patents. That's an important distinction. They're method of use patents, pharmaceutical compositions in some cases. So the precise crystalline salt form of psilocybin that they're administering, things that, you don't have such strong claims on those usually.
I remember something from the original pitch where you said you're going to chase the NCEs, but you also got a better way of making the psilocybin from yeast that's actually...
Yes, so that is a part of our plan that has been abandoned. And part of that was because we had a third collaborator and things didn't work out. But also we had some misgivings about that, truthfully. Like, as we dug in more and more, it's sort of a solved problem. And there are others who already have patents on using microbial systems to produce psilocybin. And we talked to one and he literally said, I'm sitting on this patent and waiting for a market to be created because, you know, there just is no market yet for psilocybin because it's illegal right now. So yeah, and like our third collaborator was basically saying it's going to cost us, you know, 400, 500K to get to the point of having a approved pathway for doing that. Yeah.
Excuse me, is
Yes.
there a reason why you're not producing cultures from actual psilocybin mushrooms?
Yeah.
You want to jump into the yeast production or not? I'm asking you if
Yeah.
it's safer to have a reliable source of the actual secondary compound in order to get the analogs that you are producing?
Yeah, that's a good question. There's a group, there is one researcher formerly from Berkeley, I believe, who's looking at using non-natural precursor substrates in sort of modified synthetic biology systems. And I think that's maybe what you're talking about. That you.
Oh, I didn't have a demo.
Yeah. So people are doing this. People are looking at both synthetic biology approaches in a number of organisms now, a number of host organisms to manufacture psilocybin using batch fermentation methods. And people are also looking, I know of at least one researcher who's got a small effort around feeding non-natural substrates to those same sorts of organisms in order to get non-natural drugs out of the other end. But it's really exploratory at this stage. I don't think it's with a commercial endpoint in mind.